Hello everyone, and welcome to our online lecture series for plant biology. Today, we're going to begin kind of a three-part little lecture series on how nutrients move through the plants and how they do so with the help of water. So while each of these next few lectures have its own distinct topics or flavor and takes, they all tie in together to put a cohesive look at overall movement and nutrition in plants. And we can't have this discussion about movement of nutrients in plants without discussing the water that helps drive all of this movement in the plant. So first, I want to show you all this image of rain dripping off of an awning. Kind of silly, but it does illustrate a lot of the properties that we're going to be discussing throughout the lecture today. What you'll note, first and foremost, is that this rain is forming pools. That's because the rain has the ability to stick to itself. The water in the rain has something called cohesion. Now, cohesion is a property that stems from the chemical structure of water, which, if you've taken chemistry before or are familiar with it, water has the chemical formula of H2O, meaning that there are two hydrogen atoms and a single oxygen atom per water molecule. They adopt a bent V shape with two hydrogens sticking off of the sides, and then the oxygen has two pairs of electrons coming off of the top. Now, because of something called the electronegativity of this molecule, well beyond the concepts we need to know in botany, but because of the electronegativity of this molecule, there's generally a negative flavor to this side of the water molecule and a negative flavor to this side of the molecule and a positive flavor and a positive flavor on each of those sides where the hydrogens are. What this causes is the ability for water to stick to itself, or cohesion. So you've got the water molecules lining up in such a way that H's will hang out next to O's of other molecules. And O's will line up near H's of other water molecules, creating kind of this webbing. You've seen the strict structure this webbing can adapt when water freezes, forming ice. But even in your glass, as water moves around in your cup of water, these hydrogen bonds that drive cohesion allow water to stick to itself. And therefore it does so when it's rolling off an awning or when it's coming out of the sky. Additionally, you'll notice that that water is sticking to the awning itself. Not only does that slight positive and slight negative charge that we see in water allow it to stick to itself, but by the property of adhesion, water can also stick to other things that have these slight charges. This oxygen is not limited to binding to other hydrogens in other water molecules. If something else comes by and has a positive charge, it too will form a temporary weak bond that can allow water to stick to it. You know adhesion very well every single time that you try to pick up a puddle of water on a table with a paper towel. The water very quickly seeps through the paper towel when you touch it. This is because the paper towel itself serves as a scaffold, a ladder, that water molecules can grab a hold of. They move through it very quickly. they grab onto the different pieces of the paper towel very quickly. After all, that paper towel is made of plant parts. The same plant parts that move water throughout the plant. So you can see how quickly and effectively our cell walls of our xylem are able to move water through the plant by using a paper towel to scoop up water from a table. Kind of crazy, right? So these properties of cohesion and adhesion drive water movement and therefore nutrient movement throughout the plant. The overall trajectory of where water is moving, where it comes from to where it goes, is illustrated by this cool little schematic from Wikipedia. The ultimate endpoint of all the water in a plant is transpiration. Transpiration is the loss of water through stomata on the leaves of a plant. And that's really weird to think about. Every drop of water that's absorbed by a plant's roots, here in step 1, in water uptake. Every single drop of water that gets absorbed, moved through the xylem in step 2, and you see the properties of adhesion illustrated here, where these water molecules are grabbing onto the sides of the cell, and as they grab onto the sides of the cell, occasionally they grab direct right, left, sometimes they grab up, and when they grab up, they move up, and when they move up, and then eventually out. The transpiration in a plant is occurring because its stomata are open. Its stomata are open so that way CO2 can come in. But because the stomata are open, water is also coming out. In the same way that when you open your mouth on a windy day or into a fan, you'll be losing water in your mouth even if you hadn't intended it. And so these concepts of transpiration, pulling all of the water through a plant from its roots to its shoots to its leaves and out the stomata, cohesion, keeping water sticking to itself while it's in the plant, and adhesion, helping drive water up the plant, are our 3 major forces that together will help move water up through a plant. You'll notice here that this movement is unidirectional. Water is leaving through the stomata and being taken up in the roots. It's very different than our veins. So even though we call plant vascular tissue, or vein tissue, our xylem and our phloem, even though we call them our veins, they are unidirectional veins. They travel in one direction. Xylem always from the roots to the shoots and out the stomata. Phloem do have some ability to move back and forth, but for the most part, phloem movement is also unidirectional. As we see in this second infographic, again, thanks Wikipedia artists, y'all rock. Here we have a potato plant with our beautiful little potato flowers and those lovely tubers beneath. We see that the shoots will make photosynthetic product, and they will transport it both to the flowers, so moving up, as well as down below to those potatoes as an ultimate sink. And so another good title for today's lecture, some of the others, could have been source-sink dynamics, because it is that relationship between moving sugar from where it is highly concentrated in the leaves, the source of all photosynthetic production, to the sink of all photosynthetic production, where you're going to store it, in the roots or in a new growth towards the shoots. And at the same time that sugar is being trafficked either towards the SAM or towards the RAM, you've got water and minerals traveling unidirectionally up the plant. Now, as a bonus question, you'll see that there are many different aspects to these roots, and you can additionally have added the fact that there would be mycelial growth, or fungi that occupy and help absorb water and nutrients for the plants. But as the illustration suggests, you've got photosynthetic product moving through the phloem, and you've got water products moving through the xylem. So let's take a moment now to actually talk about phloem, because we've mentioned it a bunch of times thus far. So now let's go over the exact cell types, because I don't think we've done that just yet. So the first of our two cell types in phloem is our sieve, make sure I'm spelling this right, tube cell, or sieve tube element. Oftentimes it's called or referred to as an element because as a cell, it lacks a nucleus. The loss of a nucleus, the loss of that super defining feature of eukaryotic cells, makes you frequently lose the label as a cell. We see the same thing occur in our xylem, where vessel elements are elements because they've lost a nucleus. They've lost everything else as well. In contrast to vessel elements that have lost all membrane-bound things in the cell, including the plasma membrane, they're just a hollow cell wall. Sieve tube cells, or sieve tube elements, have lost the nucleus, have lost their vacuole. They've even lost ribosomes, so they're not making a whole bunch of new proteins, but they still do maintain the plasma membrane, and very importantly, they have lots and lots of endoplasmic reticulum, or ER. That's because the endoplasmic reticulum is the membrane-bound structure in a cell that's going to help that cell move sugar around. Whereas the vacuole is designed more specifically for water control, the ER helps plant cells move around sugars. So it makes sense that a cell that's designed to move around sugar would be primarily focused on having a ton of endoplasmic reticulum. Now, the reason why the sieve tube cell lacks a nucleus and has lost its vacuole and its ribosomes is because you want to hollow out as much space, minimize obstacles for sugar, for the movement of solutes throughout the cell. Therefore, you get rid of every piece of furniture that isn't directly involved in moving sugar, like the nucleus, like the vacuole, like those ribosomes, and keep that plasma membrane and endoplasmic reticulum so that way you can really maximize sugar and mineral movement. Now, our other type of cell, and these two are paired, is called a companion cell. A companion cell is kind of the opposite of a sieve tube element. Whereas a sieve tube element is large and bulky, so that way it can move lots and lots of sugar, a companion cell is generally fairly small in size. And instead of having no nucleus, it is a cell that is mostly nucleus. It has lots of ribosomes. So the pieces that your sieve tube element lacks are expressed twofold in your companion cell. The companion cell has a large nucleus, has lots of ribosomes, has lots of mitochondria, so that way it has a very fast and strong metabolism, lots of energy when needed. And then, as a sign of its commitment and devotion, it has its plasmodesmata, or cell-to-cell connections, to only one sieve tube element. So I like to caricaturize our sieve tube element and our companion cell as a cartoon of the Frankenstein's monster. The sieve tube cell is an absolute behemoth. It is strong, it is big, it is the monster. No brains, not going to think much, just going to lift and move sugar around. In order to get its orders, in order to listen to its commands and properly be able to function, it needs an attached brain. A small cell, can't move very much, but it's very active with its tons of mitochondria and ribosomes, and does the thinking for our sieve tube cell, our sieve tube element. So you've got this pair of the brawn and the brains of our phloem. And as the story of Frankenstein, in particular, the Frankenstein monster itself, is one of my favorite Halloween costumes and Halloween stories, my brain naturally goes to this comparison of something that is strong and does the heavy lifting, and a quiet thinker in the back pulling all of the strings. So those are our two major cell types in our phloem. Now you do have some support cells. They're not going to be your companion or your sieve tube, but you might see some parenchyma in the phloem. So these would be cells that are really good at storing sugar for a bit, some metabolic function. They're just general living cells needed to provide the energy to their neighbors because, hey, guess what? Even these cells without a vacuole still need regular supplies of water. They still need regular supplies of energy. And they get them from some of their nearby neighbors of parenchyma. But beyond metabolic support with parenchyma, you've also got structural support cells. Because remember, without an internal skeleton, plants need structure in many different places throughout the organism. That structure derives primarily from two different types of cells commonly found in phloem. The first are fibers, like the very thick fibers used to create this cool mask, and then sclerids. These irregular cells that provide structure to nearby storage cells, like we see in this banana, where one sclerid in the middle has attachments from lots of storage cells, allowing these to grab on and be supported, giving you that major banana shape. But also, that sclerid gives bananas its wonderful texture. In fact, sclerids are largely the reason why you can add a frozen banana to milk, blend it up, and it comes out much the same as a milkshake, as though you added ice cream to it. It's because you get that thickening consistency from the tissue of the banana, from the cell walls of these sclerids. The more you know, right? And I'm a big fan. Summertime, frozen bananas in a milkshake, the best, right? Add some fresh strawberries to it. Something my parents did for me when I was a kid, and I continue doing it to this day. Another structural feature of our cell walls in our phloem is that they will frequently have the addition of a sugar not always found in other parts of the plant. And this is because this sugar is oftentimes used around those plasmodesmata. It's used to strengthen cell-to-cell connections. And you best believe that if you're moving a whole bunch of sugar throughout all these cells, these cell-to-cell connections at structures like the sieve plate better be supported. They better be strong. Your plasmodesmata need to be reinforced. And that sugar is called callose. In fact, you'll oftentimes hear it referred to as a callose plug because they're oftentimes laid down between cells, helping them connect stronger. We also see callose in reproductive tissue, like pollen tubes, which we'll be looking at later this semester, probably in lab. But it's interesting to note that there are differences in the cell walls between our phloem and our xylem. Plasmodesmata are incredibly important in phloem. Callose is a polysaccharide that helps reinforce those plasmodesmata. Therefore, that gives us another way to differentiate those features of xylem and phloem under a microscope with different stains. Kind of cool, right? So now that we have a good idea of the cells involved in our phloem transport, and we know the directionality of our transport from uptake in the roots to transpiration in the shoots, particularly the leaves, let's take a moment to talk about the actual physics of what's going on in a plant to move water throughout it. Because it's kind of crazy that plants move water throughout them without expending any energy. Water movement is free. Flow movement is fairly cheap, but water movement is free. Think about how different that is compared to humans. Your heart has to be beating regularly in order to move water throughout your body. You spend so much of your life and so much of your energy and so much of the food you eat to move water throughout your body, so that way every single part of your body has water when it needs it. A plant is doing the exact same thing without expending any energy. It's absolutely nuts to me. So again, this process is illustrated very well through this Wikipedia diagram as our different source-sink relationship. The other thing that you need to keep in mind when we're discussing this diagram is that where in the plant are our xylem and our phloem? Well, as we've said multiple times, either in our secondary growth lecture where we talked about tree trunks and the fact that the xylem, the water transport stuff, is in the middle and the phloem is on the outside, or whether we talked about stem cross-section or the root apical meristem or the shoot apical meristem, it's always going to be xylem in the middle, phloem on the outside, and they will be bumping up against each other. That is critical for this discussion because water is able to move up through the plant in part because those xylem features are adjacent to phloem. So why and how does that work? Well, from the beginning, you've got your source of all sugar in the plant, all the sucrose. This is just another type of sugar in the plant. Table sugar is being made in our leaves. It is loaded from our cells in the leaf into our companion cells, and our companion cells then traffic it into our sieve tube elements, or our sieve tube cells. And then it gets moved down the plant slowly but surely up until it comes up to companion cells in the roots, gets moved in the companion cells in the roots, and it's ultimately stored in the sink or the root cell in a way that is no longer water soluble. Whoa. Why did I bring that up? Why does solubility matter here? Why does the fact that the sugars down in the roots are stored in such a way so that way they don't affect water dynamics? We're not even in the xylem, right? It has everything to do with that water transport. Because all of this movement is being driven by a force called diffusion. Diffusion is a fairly simple thing to grasp. Where things are in high concentration, they will move to areas where they are in low concentration. That means that this is the same principle whereby you smell perfumes. If I spray them on the whiteboard at the front of the room, it will then travel slowly but surely to your nostrils because there's a ton of it concentrated on the whiteboard and very little of it in your nostrils, if any at all. It will diffuse in that direction. In the same way, where sucrose is being made, transported into companion cells, you have a lot of sugar in those sieve tube and companion cells near the source. As you start to move it down the plant, you get a little bit less, and then even less as you go down up until you find where it is stored. Now, all of the sugar from the source, the sucrose, is stored in such a way so that way it dissolves in water. And when it dissolves in water, it can affect the diffusion of things. Think of it in the same way like a perfume that's aerosolized and sprayed and moved around the air versus a brick of wood. The brick of wood isn't moving anywhere. Just because the right side of the room doesn't have as much lignin as the left side of the room, that brick of wood is not getting moved around. It's not soluble in the material that's moving around in the room, the air. In this case, the material moving around is water. Sugar or sucrose, like your table sugar, dissolves in water. If you've ever made Kool-Aid, you've seen that principle firsthand. But by the time it gets to the roots, that sugar, or rather when it is finally stored in the roots, that sugar is converted from sucrose to starch. Something akin to the sugar that you find in potatoes, right? By storing it in a non-soluble form, you are not messing with the diffusion of the substance throughout the plant. It is now stashed away in a box so that way it doesn't affect this gradient of going from places where there's a lot of it to places where there's less and less and less. So that way, every single bump down, let's go ahead and say that you start with 10 parts per million of sugar at the top, then it'll have 9 parts per million a little bit further below, 8 parts per million, 7 parts per million, 6 parts per million, 5 parts per million, 4 parts per million, and so on and so forth. These are completely made up numbers. But because of that gradient, sugar that's currently sitting at 10 parts per million goes, hey, there's 9 parts down there. There's more of me down there. There's less of me down there. Let's try to equalize this. Oh, there's less of me even further down below. There's less of me even further down below. There's less of me even further down below. It's the same reason why if you take a drop of food dye to your glass of water, the food dye just doesn't stay concentrated in the center. It starts to split out where there's less of it. Now this applies to our sugar in our phloem. But at the same time as there's this gradient of 10, 9, 8, 7, 6, 5, 4 on the sugar side of things, you then have to look what's happening in the adjacent water-carrying vessels. Now keep in mind that this is 100% water, right? The idea here, moving through the xylem, is that there's nothing but water. But because they are adjacent to the phloem, the water in the vessels aren't just going to want to stick to our vessels. They want to move around to where there's less water in comparison. So we'll switch the ink that I'm using here to look at the fact that if you have 10 parts per million of sugar, that means you'd get maybe, let's make this add up to 100 to keep the math consistent. You get 90 parts of water. And then down here you get 91 parts of water, 92 parts of water, 93 parts of water, 94 parts of water. And so you've got this gradient where there's less water up the phloem. So what does the water in the xylem want to do? Well, if it's at 100% here, it's going to want to move over there, but it's also going to want to move up. It's going to want to move a little bit diagonally. And it's going to use the property of adhesion to those cell walls in the xylem to help use it as a ladder. So it's going to climb both a little bit to the right and a little bit upright. And then again, a little bit upright and a little bit upright. And in this movement or pull to keep tracing where there's a little bit less water so that way it can help equalize across those two, you've got the movement of water in the vessel for free. Not only is transpiration help pulling the water up by the property of cohesion, because remember, the water is being pulled out of the stomata of the leaves through cohesion, but you've also got adhesion and diffusion working to pull the water up. And think about how forceful this is. It's beating gravity. I'm going to say that again because it's so profound. The forces in xylem allow water to beat gravity without using any energy. It all happens for absolutely no metabolic cost to the plant. It's happening for free. So let that sink in for a moment. This whole beautiful source-sink relationship. In a bit of a simplified cartoon, I'll draw it out again. You've got lots of water. Whoops, that's lost. You've got lots of water coming in from the roots, and then it's being pulled up by cohesion to the stomata. But at the same time, the xylem is adjacent to our phloem. And our phloem has lots of sugar near the source of all sugar in the plant, and then less sugar in the sink, the roots of the plant. Now, phloem movement costs energy. It's not free, but it just so happens that you're moving sugar, and guess what? Sugar is the currency of energy, so it makes it pretty cheap to do all of this, since you're literally moving energy, helps move itself. But you're moving it in incremental steps, so that way there is a concentration gradient established in the phloem. So that way you go from lots to a little bit less, 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 till you've arrived at your sink, where that sugar will be stored as starch or amylose, a sugar that does not dissolve in water and therefore does not affect diffusion. It's now taken out of the system. And we just so happen to love starch. I'm a huge fan of potatoes and French fries, and that's what they are, large sources of starch. Now, at the same time that you've got this lots to less movement in the phloem, it's also driving the movement of water up the xylem, because water is going to move in places where it has high concentrations, like here, to places where there is less water. But as it turns out, the more sugar you have, the less water there is in that region. So you've got this continuous ladder that water can use to help drive it up, constantly be pulling it up and actually beating the force of gravity. I would encourage you to try to explain this yourself. Pause the lecture, draw it out, make sure you understand the forces moving here. I'd recommend you include some fake numbers to help drive this process home, since that really, to me, cements it. Even if you are not mathematically inclined, it does help make you understand how the concentration gradient change can really drive the movement of water and sugar throughout our xylem and phloem. So that's the major reason why things are moving throughout the veins of the plant. Moving through the xylem for free due to diffusion, and moving through the phloem due to variations in concentration gradient by the source-sink relationship from leaves to roots. Now, it is through this whole movement of water and sugar that you will be transporting nutrients and everything that you need as a plant in order to live. So what happens? Here are two random vocabulary words that I think are important, but I really couldn't find a great place to put them, so here we go. So what happens to a plant when you cut the phloem? Well, we just spent a long while talking about how phloem is actually a major driver of the movement of water throughout a plant. Without phloem, it's a lot more difficult to transport a whole bunch of water. So if you cut the phloem, you actually kill the plant. Not only is it unable to now transport sugar to its roots, but it's also not really transporting water as efficiently. This process of cutting the phloem is a way in which we kill a lot of trees when we no longer want them on our property. We call this girdling. That's where we cut the tree trunk just deep enough underneath the bark to cut a line in the phloem. Not cutting the tree the whole way across, but just cutting a straight ring around it, so that way no phloem to phloem connections are made across the entire tree trunk. This really limits the amount of water that can be transported up to the leaves, and therefore the leaves start drying out and dying, and also limits how much nutrients can get to the roots. And so the process of girdling is one way in which you can kill trees, or anything with phloem, to be honest. For another random, almost Jeopardy-like association with this lecture, is that there are organisms that are able to puncture into the phloem of plants to steal that sugar. They live on sugar water alone. Things like aphids. These are insects that are able to puncture into the phloem and steal the sugar and minerals that are being transported within the phloem. That's right, we will be talking about the minerals that get transported through the phloem, because there are things outside of just sugar being moved around there. But aphids are able to sneak in and steal it. Now, this matters a lot if you actually want to be a plant physiologist studying what is in the phloem, because for a long time, folks couldn't figure out how to get into the phloem without damaging the plant. We could try needle after needle, but a plant would just seal up the puncture wound from the needle and actually block us from pulling anything out of the phloem. So none of our needles were working. So a clever scientist thought, hey, well, there's this pesky little insect called an aphid that's able to puncture into the phloem without causing it to seal up. In fact, they drink so much of the sugar water that it starts coming out of their butts, and they have little drops of sugar water on their rear ends because they're just drinking up so much of the phloem syrup. Well, aphids are able to do that through their specialized needle-like appendage that punctures into the phloem. But after the aphid has injected itself and its needle-like appendage into the phloem, the scientists then thought, hey, well, guess what? Your giant tongue-like thing is in the phloem. Now I can rip off the rest of your body and leave your tongue-like thing in there. and therefore anything in the phloem will come out the other end. It was supposed to be going into your mouth so that way you can eat it. But if I rip off the rest of your body, now it's just coming out of the needle-like thing. And this is actually the way in which scientists started studying the contents of phloem. We could get directly in there because aphids had evolved in order to suppress that blocking reaction that occurs in plants, that defense reaction. When something cuts the phloem, they seal it out. But aphids get around that, and then we got around it by removing the rest of the aphid body that would normally eat the phloem sap, and then collecting the phloem sap for ourselves. Thanks, little bugs. You guys wreak havoc on my garden, but I think we'll call it even after that one. There are some other things, and this class won't really focus on them in terms of, or at least for now it will not. I'm sure in the next 20 years of scientific research, this will become a more important to plant biology. But as of our current time, it's not widely discussed, or perhaps it'll be discussed more in a future plant biology class, like more advanced rather. But other things that move through the phloem, other traffic. This is, of course, besides sugar and nutrients. Are molecules called small RNAs. These are signaling molecules. So stuff like stress or defense can be moved throughout the phloem in the form of small RNAs. Funny story about that is I actually had a professor in graduate school who, despite the fact that this is a well-established field of research, just didn't believe it existed and marked me wrong on one of my exams. I was totally confused as to how I was supposed to answer that question when his question was literally identify the molecules that move through this phloem, and I mentioned small RNAs, and he said, yeah, I know it's out there, but I don't believe it, so you're wrong. All right. Talk about a little bit of cognitive dissonance, inability to recognize the entire field as probably true. Up until this point, still to this day, there's no evidence in the contrary that all of that field of research is wrong, so we're still standing by it, and I feel a little bit awkward about that graduate level class. And then the other thing that's moving through are some occasional proteins, and a lot of work is being done on this because, in particular, flowering signals. Flowering proteins are able to move throughout the phloem, and that's something that we're studying more and more. So, more to come on that topic. But we do get some signaling molecules in addition to the sugar and nutrients. So in that way, these act as kind of hormones for the plant, right? You've got hormones flowing through your bloodstream whenever you need an adrenaline rush. Let's say a tiger pops up behind you and you need to run as fast as humanly possible. It is adrenaline that goes through your bloodstream to instigate the rest of your body to go into ultra panic mode and please, please, please let me run faster than the individual next to me. In the same way, plants are able to signal things like sometimes stress, but more often flowering signals throughout their phloem in a similar way that hormones act in you. So now we're getting to the plant nutrition part of this lecture. The first part discussed how soils and sources and sinks drive the movement of water and sugar throughout the plant. And now we'll talk about plant nutrition and the nutrients that are essential in helping a plant grow. An essential nutrient is a nutrient that you must have in order to complete the life cycle. This means that the plant uses it in its metabolism, uses it in its body, and if you don't have enough of this, the plant will not produce flower and fruit and make babies. It's essential to the growth of the plant. Now, in terms of these essential nutrients, you have those that you need a lot of, our macronutrients, and those that you need very little of, our micronutrients. In terms of macronutrients of a plant, this is where just having these notes forever helps. The major macronutrients for a plant are sulfur, phosphorus, magnesium, calcium, potassium, and potassium, I will ask that y'all keep track of its chemical symbol, K. Nitrogen, whose chemical symbol is N. Oxygen, carbon, and hydrogen. I list those three together because the oxygen is derived from water, the hydrogen is derived from water, and the carbon is derived from the air. These are the major backbone of sugar. So yeah, duh, they're needed in large amounts because everything in a plant is made of sugar, or pretty much everything. So those are your macronutrients, things a plant needs a lot of in order to grow correctly. In terms of your micronutrients, they are commonly molybdenum. We haven't talked much about it, but a plant does need it in order to grow, to make some of its proteins. Nickel, same deal. Copper, super important in different proteins. Zinc, that one is very essential to protein structure. Manganese, you already know where manganese is absolutely critical for. Think back to our photosynthesis lectures. Manganese was an important component of the oxygen evolving complex in photosystem P680, the reaction center. Without manganese, the reaction center doesn't operate. Without a reaction center, photosynthesis can't happen. You don't need a lot of it, but you better have some of it. Boron is another common micronutrient. Plants need a little bit of it, both in membrane structures as well as proteins. Chlorine, lots of ion signaling in chlorine. And then last but not least, iron. Think back again to photosynthesis in our iron-sulfur proteins. Iron is needed in small amounts in order to complete photosynthesis. It is a cofactor in photosynthesis. It's needed to be there in order to facilitate the process. Now, in terms of some other nutrients that we'll be talking about in some more detail, for our macronutrients, sulfur is needed both in proteins and DNA. So this is kind of just some other facts about these different macro and micronutrients to help keep track of why they are macro and micro in the first place. Sulfur is important in building our proteins and DNA. Phosphorus is incredibly important for energy in DNA. Need lots and lots of phosphorus. Magnesium, again, proteins. Calcium, lots of signaling. Potassium is going to be important in stomata function. So you will not be able to close your stomata unless you have a sufficient amount of potassium. But then also potassium is key in water movement. One of the driving elements in the phloem that help move water up through the xylem is the presence of potassium. So you really need to regulate how much potassium you have as a plant. And then nitrogen, well, that's kind of needed for everything, including chlorophyll. Major component there. Can't make chlorophyll, can't perform photosynthesis. So plants need lots and lots and lots of nitrogen, plus your DNA also needs nitrogen, plus your proteins need nitrogen. Nitrogen's just everywhere. In fact, one of the most common fertilizer mixes out there, whenever you're trying to plant a field, if you're farming or working in your garden, you'll see fertilizer mixes that address your NPK levels, your nitrogen, phosphorus, and potassium levels, because most often these are the macronutrients that are lacking in our soil. Nitrogen is incredibly scarce, and we'll end our lecture talking about why. But the same also goes for potassium and phosphorus. Our agricultural plants that are oftentimes growing quickly in high demand in a short growing season, well, we want to make sure that they don't want for nutrients, so we fertilize them. We give them a very happy environment in which to grow, and therefore they grow quickly for us. NPK fertilizers are a very common way of addressing those macronutrient needs. Now, what happens to a plant if it lacks nutrients? So what happens when there is a shortfall? Well, any kind of lack of nutrients or malnutrition in a plant, or, oops, that's misspelling nutrition. No, we spelled nutrition correctly. Malnutrition in a plant is actually going to cause developmental problems. Meaning that the plant might not grow as tall or grow as strong or have a slightly weakened immune system, etc., etc. In the same way that malnutrition can mess with human development. For example, one of the scariest statistics out there, I think, is a comparison of height in North and South Korea. South Koreans generally do not have malnutritional issues. North Koreans, unfortunately, frequently do. And despite the fact that these two groups are genetically fairly homogeneous, meaning that their genes say they should be about the same height, you have a noticeable difference in several inches of the average height between a North and a South Korean. North Koreans being shorter. Yikes! Malnutrition in the modern world. Still happening. Definitely a major bummer. Malnutrition in plants can lead to a number of different developmental issues, especially as certain lineages of plants have different nutrient needs. Not every single plant needs all of those micro and macronutrients in the same amounts. In fact, even comparing things like our eudicots versus our monocots, so things like grasses versus things like grasses versus things like cherries, you will get different macronutrient needs. Eudicots are generally recognized as needing a lot more calcium and boron than our monocots. And monocots are variable enough that there isn't a general trend across them, but they do grow fairly quickly, and their nutrient needs are still as important as other plants. But generally speaking, when you're looking at your eudicots, if you're working on something like a cherry orchard or an apple orchard, you're additionally monitoring calcium levels and boron levels, and in particular the pH of the soil, in order to make sure that it is not messing with the absorption of nutrients through the soil. More on that later. But in terms of the developmental issues that you will see in a plant, there are, generally speaking, 3 different categories of developmental issues. The first is stunted growth, meaning the plant is smaller. The second is necrosis, which is certain cells in the growing leaf just dying. So you can imagine that a plant is growing, it lacks nutrients, and because it lacked a nutrient when a cell was starting to mature in the region of maturation, it lacked those nutrients, cell couldn't finish growing, it just dies. So you get a lot of black spots in this kind of phenotype. And then last but not least, we have our chlorosis. And chlorosis is the yellowing of leaves, meaning that they're lacking the materials necessary in order to perform all the metabolism associated with photosynthesis. They're not building your chlorophylls correctly. So what do these actually look like in a plant? Well, there's some variation to it actually. Stunted growth you recognize as the fact that the plant is not growing quite as fast. It's just a shorter plant. But with the necrosis and chlorosis, the dying of cells or the yellowing of tissues because of inadequate materials to perform photosynthesis, it differs based on the different nutrients that might be lacking. For example, you can get necrosis in different spots in the cell leaf. Or when you're dealing with necrosis due to the lack of potassium, you might see necrosis in the leaf margins or the edges of leaves, where they're expanding the most in either this legume or this grapevine. You see necrosis in those leaf margins because the plant is lacking the potassium necessary to expand that leaf, the ions necessary to help regulate that growth at the margins, which are expanding quite quickly. And therefore, those cells, instead of growing shorter or whatever, they just die. They lack all the nutrients necessary. Chlorosis can also manifest in different ways. Here on the left, we have a very chlorotic corn plant. In fact, despite the fact that this is annotated as an example of chlorosis on Wikipedia, I'd even imagine that it could be a mutant whereby chlorophyll is not expressed. That does happen occasionally. But I'd trust the farmer to know more than my backseat driving in this case. Perhaps that spot was highly chlorotic and the corn plant was just not able to make any chlorophyll. Versus the corn plant on the right. Has enough materials, does not have enough materials in order to perform photosynthesis, in order to make chlorophyll. You additionally can get like intravenous chlorosis, like we see here in this leaf, where as you get away from the veins, everything is turning a much more yellow color. The yellowing because it's not able to make the chlorophyll pigments. So while there are a lot of nutrients that cause each of these in different ways, I'll ask y'all to keep track or give y'all examples of each one of these with only one. A common cause of stunted growth, a common nutrient that's lacking and causing stunted growth is calcium in plants. Calcium being one of those macronutrients necessary in high quantities for plants. A common source of necrosis in plants, in addition to the potassium that was shown below, is phosphorus. So you can get either potassium or phosphorus causing necrosis. And chlorosis, because that nitrogen is necessary in the formation of chlorophyll, Nitrogen is a common reason for chlorosis in plants. And hopefully, based on this, it should be readily apparent why fertilizers are oftentimes good mixes of N, P, and K, with other fertilizers, or the secondary ingredients, commonly being things like calcium. Bam, right? We'll end today's lecture, because I've been going on for a bit here, by discussing mobility versus immobility of nutrients. Now, nutrients that are mobile can be recycled. What this means is that if a plant is growing and it does not have enough of a particular nutrient, it can kill off old leaves so that way new leaves can grow just all right, grow just fine. Mobile nutrients can be moved through the phloem from old leaves to new leaves. This means that old leaves die or show symptoms of malnutrition, whereas new leaves are okay. So mobile nutrients sacrifice old leaves to help benefit new leaves. In contrast, immobile nutrients cannot be recycled. They're stuck in the places they were built. Therefore, this means that new leaves will oftentimes show these symptoms first. If you don't have enough of it in the soil and therefore in the phloem of the plant, it's not going to be able to get to the shoot apical meristem where it's needed in high quantities, and therefore new nutrients or new leaves will be screwed up because they do not have these immobile nutrients. As an example of some of our mobile nutrients, you've got N, P, and K. They frequently will be recycled from old leaves to new leaves. Not that the old leaves couldn't use them, but rather that the plant is sacrificing the old for the new. In terms of immobile nutrients, the most common one is, or rather, not, I shouldn't say the most common one, but the most commonly cited in plant biology classes is iron, or Fe, as well as manganese. And I point these two out as our immobile nutrients, in particular because they're both essential components of our photosystems. Manganese being present in the oxygen evolving complex that cannot be recycled, and iron being essential to a lot of the different steps in photosynthesis, particularly in those iron-sulfur proteins. Those also will not be recycled. And so that's where we'll end off our lecture today, hopefully by drawing a connection between water forces, like cohesion, adhesion, and transpiration, to how that helps move water through the xylem while also moving sugars through our phloem, thanks to the help of our companion and sieve tube cells. And then giving y'all a little bit of a discussion about plant nutrition by going over our essential nutrients, our essential macronutrients, our essential micronutrients, and what happens to a plant when they don't have those nutrients. Do they suffer stunted growth, necrosis, or chlorosis in new leaves or old leaves? See y'all next time for a continuation of this topic, where we take a closer look at our xylem.